This is the fourth and last part of today's introductory session in cognitive neuroscience. Here we will be talking about the basics of neural activity, for example, how action potentials come about and other related neural processes, which will be important in order to understand the neuroscience methods in more detail that we will be talking about in the next couple of weeks. The universal means of communication in all animals is the nervous system, and at the very core of the nervous system is a neuron. Here you can see a neuron. You can see the dendrites, which are used to receive input from other neurons. You have the soma and the main body of the cell. You have the axon, which transmits an action potential or graded potential to synapse, which can then release neurotransmitters. And to make the whole thing fast enough, we have these myelin sheaths that are separated by nodes of Ranvier, so that these action potentials can actually be run faster. So the origin of all neural activity is the action potential, and in the following we will be talking about how an action potential is triggered. First of all, we need to think of the neuron as polarized brain cells. So the inside of a cell membrane is about 70 millivolts more negative than the outside. This voltage we can measure here is in its resting state minus 70 millivolts. So in order to understand how an action potential comes about, we need to focus on two electrochemical forces. The first one being the electric potential. Here's an electric potential, the left-hand side, because there is a positively charged ion on one side of the membrane and a negatively charged ion on the other side. So the positive one is attracted by the negative one, but at the moment they can't trespass here. Here on the right hand side you can see a higher electric potential compared to the left side, because there's three negatively charged ions on one side and one positively charged ion on the other side. So this means that the difference in the charges is more pronounced, which means that the positive ion is attracted more towards the other side than here. The second electrochemical force we need to understand is the diffusion force. So this is simply put how much force there is to push particles of the same kind apart. So here you can see there's 5 particles on one side and no particle on the other side, which means they will be driven to go to that side, but again at the moment they can't because there's this membrane in between. On the right hand side you can see a higher diffusion force because the difference in particles on one side and the other side is even larger. So they're driven even more towards this side. Okay, let's talk about the resting potential. So this is when nothing is really happening in a neuron, but actually something is happening to keep the resting potential stable, and how that works I will explain in a second. Let's zoom in on an axon here. You can see extracellular space, the cell membrane is here, and this is the intracellular space. So there's a lot of different particles and ions inside and outside of our neuron, but I will be focusing on organic anions, potassium and sodium, because they're most crucial in order to understand how the resting potential is kept stable and how an action potential comes about. So the organic anions are negatively charged. So basically they, because the outside of the cell is more positive than the inside, they want to they're driven by the electrical and the diffusion force to the outside. Why is that? So you can see the inside is negative, they are negative, so they want to go to the other side, which is more positive. That's the electrical force. At the same time, there's also diffusion force because there's more of them on the inside of the cell than on the outside of the cell. So they work both in the same direction, and you might think, okay, maybe they can get there, but they actually cannot because there's no channels for them to go through the membrane. So they have to stay inside. For potassium, which is positively charged, we see a bit more of a complicated picture because the diffusion force, this purple arrow, drives them outside of the cell because there's many of them on the inside and only few of them on the outside. So they want to get out. But at the same time you have an electrical force keeping them in, going to the opposite direction. This red arrow indicates that. Because the inside is negatively charged and they are positively charged, so by staying inside they basically go into the direction that is determined by the different charges. So we have opposing forces here, diffusion force outside, electrical force inside. So what happens is, because we have K plus potassium leak channels, that means they can actually go outside until they reach an equilibrium, which is happening at minus 84 millivolts for K plus. So this means at this point the two arrows are equally long, indicating that the diffusion force and electrical force are in line. Okay, the last thing we need to talk about is sodium, Na+, which is also positively charged but larger. We also have Na+ leak channels, and as you can see, the diffusion force and electrical force are again going into the same direction. Why? Because, as I said, Na+ is positive, so it wants to go inside of the cell, which is more negative, and at the same time there's also more Na+ outside of the cell compared to inside of the cell. So again, both forces working in the same direction, and there's Na+ leak channels, but they are, because Na+ is a bit larger, it has more trouble going inside, which in practicality means that it takes longer. There's just less of an influx per time compared to K+. But they can also reach an equilibrium, which for Na+ would be at 66 millivolts. So at 66 millivolts plus 66 millivolts, this is when Na+ is at its equilibrium, meaning that the diffusion force and the electrical force would be at zero both. But as I mentioned earlier, we have a difference of minus 70 millivolts between the inside and the outside. So how does that come about? It means that first of all, since this is not at the equilibrium, that there's still some kind of force driving potassium out of the cell rather than inside of the cell. And even though there is some Na+ going inside of the cell, because we are at minus 70 millivolts, there's still a force driving Na+ inside of the cell. So what do we do in order to keep that balance? Well, we have this ion pump here, and the ion pump does the following. It takes 3 Na+ from inside the cell, pulling them out in exchange for 2 K+ ions. By that means we will never reach the equilibrium here, or this equilibrium, but rather stay at minus 70 millivolts. Now let's have a look at the graded potential. In order to understand this better, we will have a look at the dendrites here. Let's zoom in here. So what we see here in this graph in a second will be how voltage changes over time when different processes are happening in the neuron. and outside of the neuron. So let's say there is a synapse that releases a neurotransmitter that can lock onto this gated Na plus channel. And then what happens, so this would be coming from a different neuron, right? So what then happens is that this gated channel opens up, allowing Na plus to go inside of the cell. Remember, in the resting potential, Na plus still wants to get inside because the the equilibrium for Na plus would be in a positive, would be a positive voltage, but we are still at minus 70. So by opening these gates and letting Na plus in, what happens is, of course, it's less negative. This depolarization happens. Depolarization means it's less polarized. The polarization being at minus 70 millivolts. So we're less negative here, depolarization. But this only happens for a short period of time, because then the neurotransmitter is gone, the gated Na plus channel closes again, and then because Na plus is distributing inside the cell, we will see a repolarization. So this is a graded potential. And on a larger scale, like for the whole neuron, this could look like something like this. We have the axon, releases the neurotransmitters, and then this allows positive ions to go into the cell body, which means there's a depolarization very strong at first here, at this point. Then it's distributing throughout the body of the neuron, which means there's still some graded potential here. And then while the graded potential slowly disappears here, there's still some propagation of the signal towards the axon here. But this is never as strong as at the point where the ions originally flow into the neuron. So it decays with time and distance. So in order to understand the action potential, we will zoom in into the trigger zone. So this is the zone near where an axon starts from the soma. And we will see what happens if 3 such graded potentials arrive at the trigger zone. So this is Na plus ions coming into the trigger zone from somewhere here. This is a propagated ion flow from a dendrite where originally a graded potential was triggered by neurotransmitters from another neuron. So these Na plus ions arrive in the trigger zone, and then what happens is that you see some graded potential here. Then it might be that at the same time there's another graded potential arriving from a different dendrite. Then we have something like this, and maybe even more. And then we have something like this. So if this many Na plus ions are at the voltage gated channel, they sum up to something that might actually reach this threshold here. And that means when this threshold is reached, that the voltage gated channel can open. This is a channel that allows Na plus to enter the inner of the cell. Please note that we're kind of zooming out. So this is the same graph, but we're at a different scale, so you can see more. Okay, so what happens if the Na plus flows in? We have even more of a depolarization, so it goes even more into the positive range of the voltage here, and this causes a second thing to happen, namely voltage gated channels again that open up at more for depolarization, letting K plus go out of the cell. Remember, K plus still wants to get out of the cell because of the electrochemical forces that in some point into the direction outside of the cell. So what happens if these K plus ions go out of the cell is a repolarization, meaning that again the inside is more negative than the outside because all the K plus is missing now. And this repolarization literally means we re-instantiate a polarization. So this, because it happens with some kind of lag, will also lead to a hyperpolarization for some time. So there's like an overcompensation, like before the voltage gated channels can close again, there will be more K plus going out, and this causes this hyperpolarization here. And then we're back at the resting potential of minus 70 millivolts. So I've shown you this for one pair of Na plus and K plus voltage gated channels, but of course there's very many of them nearby each other, and then we can see this kind of cascaded effect. So Na, we have a positivity here which opens this channel, more positive ions flow here, which means that this Na plus channel opens as well, more Na plus goes in here, which changes the voltage gated channel to allow more Na plus to get inside of the cell. And then with a lag you will also see, I didn't animate this here, but you can imagine that with a lag the K plus channels will also open, and then the K plus will flow outside, leading to the repolarization. So what this means is that we see an action potential traveling along the axon. So again, to zoom out and get a better idea of the big picture, let's say we have a positively charged ions arriving at the trigger zone, opening of the voltage gated channels, meaning this is the start of the action potential. There's no action potential yet here, it's really just happening here at the trigger zone at the moment. But because of this positivity arriving here, the next neighboring zone will also cause action potential here. And then this goes on and on and on and on. And in the end, instead of letting more ions flow in, there will actually be an opening of channels that allow neurotransmitters to exit the synapse, and then it possibly triggering a new action potential or graded potential in a neighboring cell. Here's an overview of the voltage changes across the scope of such an entire action potential. So we start at minus 70 millivolts, the resting potential. There's a graded potential, which in this case reaches the threshold for an action potential to happen. And then we see this the depolarization here happening. This depolarization goes into the positive area, so now the inside of the cell is more positive than the outside. And then the repolarization happens, which means that a lot of positive ions float outside of the cell, leading to the resulting in a more negatively charged inner part of the cell compared to the outside of the cell. This hyperpolarization is for a brief moment. There will also be even more negatively charged inner part of the cell than outer part of the cell, so it goes beyond 70 millivolts and then back to the resting potential. So what you can see here indicated in different time windows is a relative refractory period and an absolute refractory period. What does that mean? The absolute refractory period means that no further action potential can be triggered at this moment. This is because the Na+ channels are inactivated. And then here during this time, this is the relative refractory period. Here a further action potential is possible, but it is harder to trigger. One reason for that is that we're more negative here, so you need to overcome, you need more graded potentials to reach the threshold. Okay, so we know how an action potential travels along an axon, and it is important to note that in neurons we have myelin sheaths ideally surrounding each axon, because that increases the speed. And the principle here is called saltatory conduction. Saltatory meaning something like jumping. And why is it jumping? Because imagine we have an action potential happening here. So there's some positively charged ions getting into the axon here. Now normally they would go into the neighboring directions and then maybe trigger an action potential here. But this is isolated. So here's a very thick membrane that prevents any exchange of ions. So what that means is the ions are going to the next node of Ranvier here, where we have negatively charged ions, and then trigger an action potential here. Then again, the inflow of positively charged ions can only go to the next node of Ranvier, and so on. So basically, by skipping a few steps, we get faster to the synapse. That's the idea of a saltatory conduction, and this is why myelin sheaths are important to reach a sufficient speed in neural processing. Here's a few principles of action potentials. So first of all, an action potential follows the all or nothing principle. That means as soon as the voltage passes the threshold, an action potential is triggered, and the size of the action potential is always roughly the same in each neuron. So the question is, of course, how can we code different information for example, how does a neuron code warm versus hot, or some pain versus a lot of pain, or bright light versus dim light? Well, this is all coded in the so-called firing rate of a neuron. So rather than the amplitude of the action potential varying, which cannot happen, there will be more or less action potentials happening. So strong inputs will lead to a higher fire rate of neurons. This is also related to the relative refractory period, because the stronger an input is, the more likely it will already trigger a new action potential during the relative refractory period. When you want to record neuron activity, there's three major ways to do that. There's intracellular single-cell recordings, there's extracellular single-unit recordings, and there's multi-unit recordings. Let's talk about these in a bit more detail. So intracellular single-cell recordings require microelectrodes that are very thin, up to 0.05 micrometers. You put them inside of a neuron, and then you can actually measure all stages of an action potential. So the resting potential, the graded potential, etc. This is what you can see here. So you measure the difference between the inside and the outside of the cell. The problem is, of course, as you can imagine, these neurons are really tiny, so it is very difficult to find a cell and to maintain the position with the electrode. Imagine that if you have a living tissue, there's a lot of movement involved. And another disadvantage is that there's also, you always cause some cell damage if you do that. Typical aim of such studies is to learn about cell properties. Second way to measure neuron activity is extracellular single unit recordings. So as you can see here, you don't need to get inside of a cell body, you just need to be somewhere nearby, and it will still pick up on the neural activity of that, electric activity of that neuron. You still have microelectrodes, they can be a bit larger, and you can isolate the activity of a single neuron if you're close enough, but you don't have a good enough resolution to see the individual stages of an action potential. Rather, what you typically see is these like spikes. No? So this is just the tip of an action potential, and you can see the change in firing rate. For example, there's a lot of action potentials in a row here. Then there's a pause. There's another action potential, a pause, and then a whole bunch of action potentials. So if you could zoom in, what you would see is something like this. The action potential. But what you end up seeing in the signal is really just these spikes. And this is often used in neuroscience to identify firing patterns. So for example, the spike frequency, if you want to compare different inputs to a cell. Then last but not least, we have extracellular multi-unit recordings. So here you go with the electrode relatively large, as you can see here in comparison to the neurons. You just put that somewhere inside a group of neurons, and then you will get the response of a large number of neurons rather than just one neuron. So this is what we call the local field potential. And the aim is to identify the response pattern of neural ensembles. What you can see here is the signal, what you can see here, is based on something like this. So this underlying firing rate of cells, which is something we cannot measure with the electrodes, is really more of a theoretical point to show how the signal, or what the signal might be comprised of. So for example, you can see here, so the idea here is really you have a bunch of maybe like 8 neurons here that have an effect on this electrode, and you can see if all of these neurons fire at the same time, like here or maybe here, you can see a relatively high amplitude, like here or like here. But sometimes they're not synchronous, they just fire relatively randomly, and then you can see the sum of the signal is relatively weak because there's not much going on at any given time point. So this can be used to really identify the synchronous firing of a neural ensemble. All right, so much about the basic principles of action potentials and related processes. And this was the last part of today's session, the introductory session, Cognitive Neuroscience. Next week we will be talking about electroencephalography, or EEG. Thank you very much for your attention.